Hey everyone, let's talk about the cell cycle and mitosis. Now, mitosis in particular is one of the most difficult cellular processes for a new biology student to grasp and learn about. Something that I've noticed is that, in general, it's not a subject that's taught particularly well on average. So, I'm going to throw my hat into the ring and try to rectify this, and I'm hoping that you will find this video to be very helpful as you learn about the cell cycle and mitosis for the first time. So first, let's cover some big picture concepts. So if you have a cell like what you see here, the whole purpose of the cell cycle is that the cell needs to make an exact duplicate of itself. So if it's going to do that, the cell is going to have to make duplicate copies of all of its organelles and all of its internal materials. So that means organelles like mitochondria, endoplasmic reticula, lysosomes, and centrosomes. You need extra biomolecules like proteins and lipids and carbohydrates. And then most importantly, the most important thing that the cell needs to duplicate is the DNA, or the chromatin, which is there in blue, contained within the nucleus. So the idea of why this duplication is necessary is because if we didn't duplicate all of these materials prior to the cell dividing, then each of the two daughter cells that are produced at the end of this would only have half the materials the original parent cell started with. You can see right away how this is not a sustainable thing in the sense that every new generation of cell is going to have half as much DNA, half as much protein, half as many mitochondria as the cell that came before it. And eventually, after multiple generations, you'll end up with cells that have barely anything to work with, and that's not the way that cell biology works. So first, let's talk about interphase, which is the longest portion of the cell cycle. This is truly a cell cycle in the sense that you can see it is a circular process, going around and around and around. So interphase itself consists of three different stages. These are called G1, S, and G2 phases. So what we're going to see here is that different portions of the duplication process are going to be accomplished in each of these different phases. Now, the other phase that you'll see here, called the mitotic or M phase, is not necessarily the duplication process. It is once the duplication has been completed, then we can go about divvying up those things into the two daughter cells. So once we get to M phase, we already have duplicated chromosomes, duplicated organelles, duplicated proteins, and now we're ready to equally divide them amongst what's going to be the two daughter cells. So let's start by talking briefly about G1 phase. G1 phase is going to involve a lot of cell growth and organelle duplication, with the exception of the centrosomes, and you'll see what we mean there in just a little bit. So you'll see a little arrow kind of tailing off to the side off of the G1 stage. So it turns out that not every cell in the human body and in a lot of organisms, not every cell is necessarily going to be going through the cell cycle all the time. So for example, nerve cells and heart cells do not go through the cell cycle at all. We say that they have permanently exited the cell cycle. So if a cell is going to exit the cell cycle, it will do so here and it will become quiescent. So what we're going to see here is that there are a number of checkpoints that have to be passed as a cell makes its way around the cell cycle. The first of these checkpoints is called the G1S checkpoint. So more or less, the purpose of these checkpoints is that the whole cell cycle process is extremely finicky, and it has to be done with extreme precision and extreme accuracy. Making even a very small mistake could lead to disastrous consequences for the cell or for the organism. So we have these checkpoints to make sure and verify that we are doing everything with extreme accuracy and precision, and if we do make a mistake, the checkpoint will be failed, and the cell division process will be paused momentarily while the cell attempts to fix the problem. So for the G1S checkpoint, what we're going to do here is the cell is going to check to make sure the DNA is not damaged. The next step in the process is going to be duplicating the DNA, making exact copies of it, so it wouldn't do to make copies of DNA that is already flawed. So we want to make sure the DNA is absolutely flawless before we go to the trouble of making copies of it. If a cell fails the G1S checkpoint, the cell is given an opportunity to fix the DNA damage, but in all likelihood, the cell is going to be forced to go through apoptosis, or programmed cell death. The next stage is called the S, or synthesis phase. This is going to be where we duplicate all of our chromosomes and we make copies of all of our DNA sequences. The other thing that kind of flies under the radar sometimes is that this is also going to be where we see the production of a second centrosome, which is sometimes also called the microtubule organizing center of the cell. We'll get a better idea of what the centrosome is there for here in a little bit, but for now, recognize that a normal cell that is not going through the cell cycle only has one centrosome. So in S phase here, we're going to the trouble of making a second centrosome, and you'll see exactly why we're going to need two of them later on. So we will see later on, like I said, that the duplication of the centrosome is going to be an important part of mitosis. These are microtubule organizing centers, and the microtubules that emanate off of these centrosomes are going to be important for latching on to the chromosomes and pulling them apart from each other when we divide the DNA into the two daughter cells. Next is the G2 phase. This is basically just going to involve a little bit of final preparation for M phase. We're going to continue synthesizing important proteins that are necessary for the cell division process. Now, we are going to see the presence of a second checkpoint here called the G2M checkpoint. And what we're checking for here is we're making sure that the duplication of all the chromosomes that we saw in the S phase went flawlessly as well. So you can kind of see a theme here. We want to make sure, especially when it comes to DNA, we want to make sure that everything is flawless. So if we detect any errors here, more than likely the cell's going to have to go through apoptosis and it won't be able to continue. So finally we get to M phase, which is going to be made up of mitosis and cytokinesis. Mitosis being the process in which we divvy up all of these duplicated materials to opposite ends of the cell, and then cytokinesis, where we basically split the cell in half right down the middle so that the divvied up materials end up in two different daughter cells. We will see a third and final checkpoint here halfway through M phase, and this is called the spindle checkpoint. And what we're searching for here, and we'll get some better details on this later, what we're looking for here is we want to make sure that all the duplicated chromosomes are being lined up exactly in the middle of the cell. If there are any stragglers that can't get lined up properly, then the mitosis process will be paused until we get a chance to correct that. Okay, so for the rest of the video, we're going to be talking about mitosis, but we want to make sure that we kind of have the big picture idea of what we're about to get into here. So let's talk about exactly what happened in S phase with respect to the chromatin that we duplicated. So for our example here, we are going to consider human cells. Humans are eukaryotic cells, meaning that these are cells that have membrane-bound organelles, and most importantly, have a membrane-bound nucleus where the chromatin and the chromosomes are stored. A human cell is diploid, meaning that it harbors two copies of each type of chromosome. Two copies because one copy came from our mother and the other copy came from our father. Humans tend to have 23 types of chromosomes, meaning that our diploid number is 46. Now keep in mind, humans have 23 different types of chromosomes, but that doesn't necessarily mean that dogs or cats or other types of animals or organisms are going to have the same type of number of chromosomes. So that number is going to be different from organism to organism in most cases. But our diploid number is 46. So here is part of the problem that I have noticed with how mitosis gets taught to students. You always kind of dance around the issue of how many chromosomes you have after S phase. So if a normal human cell is diploid and a normal human cell is supposed to have 46 chromosomes, that means After S phase has occurred, that cell is going to have 92 chromosomes. It takes on what is called a tetraploid property. It's no longer diploid, which is 2n, it is tetraploid. It has 4 copies of each chromosome instead of 2. So we've got 92 chromosomes to work with after S phase has wrapped up. And you can see why this has to be the case, because once the M phase is over with and we now have 2 daughter cells, we want those 2 daughter cells to be diploid. We want those 2 daughter cells to have 46 total chromosomes. So if we have a big old cell that has 92 chromosomes and then we split it into 2 cells that each have 46, that is congruent with what we want to happen. If we go into M phase with 46 chromosomes, then you're going to end up with 2 daughter cells that each have 23 chromosomes. That is not the goal of mitosis. In a separate video that I'm going to make, you'll see that that is the goal of meiosis, but it is not the goal of mitosis. Okay, so during the interphase process, which again, that is where any given cell is going to spend the vast majority of its time, the chromatin is going to be contained within the nucleus, and it has not fully condensed into visible chromosomes. So if you were to look at a cell under the microscope and you were going to stain the cell with something that would make the DNA visible, this is more or less what it would look like. You'd basically just see a big kind of circular egg-shaped haze of blue in there. So you can't make out any of the individual 46 or 92 chromosomes because that's what it looks like. It's basically just a whole bunch of spaghetti that is indistinguishable from each other. So as we are about to enter into mitosis, we are going to address 2 major logistical concerns. Number 1, there's an issue with the chromatin itself. So the chromatin as it is during interphase is going to be way too difficult to work with. It's going to be very difficult to separate a whole bunch of strands of spaghetti away from each other. So that's why as we move into mitosis, we are going to see the chromosomes condense into discrete structures that can actually be physically separated from one another. And then number 2, the nucleus, which is a membrane-bound structure, prevents the DNA from escaping out into the cytoplasm of the cell. And that is going to prevent us from being able to separate the chromosomes into 2 different sides of the cell. So we're also going to see the nucleus start to break down and disappear so that the chromosomes can be captured by the 2 different mitotic spindles, which are produced from the centrosomes that were duplicated during S phase. So you can actually see the 2 red-colored centrosomes here in this picture of an interphase cell that is just about to start the mitosis process. Okay, so the very first step of mitosis is called prophase. It is observable and distinguishable because you can see condensed chromosomes, so you no longer see that haze of DNA. You actually see the DNA take on these discrete chromosomal structures here, but the nucleus is still there. The nuclear envelope is beginning to dissolve and disappear, but there's no doubt the nucleus is still there, and the chromosomes are still trapped inside. Like we understand already, we have 2 centrosomes at this point because this is happening after S phase. And what the centrosomes are now doing is they are each migrating to opposite ends or opposite poles of the cell. So we want those centrosomes to be on opposite ends of the cell, and they are not going to do so until the next stage after prophase. Now, I do want to make a quick note about how I am illustrating this process here. So if this were a human cell, as I said before, at this point we would have a total of 92 chromosomes to deal with. 1 chromosome is one of these single structures that you see right here. So each butterfly structure that you're looking at right here is actually 2 different chromosomes that are latched onto each other. Those are called sister chromatids. It's 2 copies of the same chromosome that are latched onto each other. And we'll talk about how we're going to separate those from each other later on. Okay, so after prophase has occurred, we are going to move on to the next step, which is called prometaphase. Now, not every textbook and not every teacher is necessarily going to acknowledge pro-metaphase. Some lectures may go straight from prophase to metaphase, but there actually is an important thing that's going to happen here at pro-metaphase that I think is important for you to understand. So by the time we get to pro-metaphase, the nucleus is now gone, as you can see, and the centrosomes are now at opposite ends of the cell. Protein complexes called kinetochores in green here are going to assemble on each sister chromatid at a location called the centromere, which is usually halfway along the length of this chromosome. Not always exactly halfway, but approximately. Now, the important thing about the centrosomes here is that they are going to start projecting microtubule fibers away from the end of the cell and towards those kinetochore proteins. The kinetochore proteins are special because they are capable of binding to the microtubules. So those microtubules are going to extend out from the centrosome, and they are going to capture and bind to the kinetochore proteins, basically meaning that the chromosomes themselves are captured by the microtubule fibers. So you can see here how, as we flip back and forth between pro-metaphase and metaphase, each chromosome in each pair of sister chromatids is captured on either side from either centrosome. The microtubules are going to then, once they capture their sister chromatids, they are going to pull, kind of like a game of tug-of-war. So each pair of sister chromatids is going to be pulled from either side. And the goal here is that we want the pulling forces from either side to be equal, so that if the pulling forces are equal, we should line up the chromosomes all exactly down the equator of the cell, like what you see here. This equator is called the metaphase plate. So by the next stage of the cell of mitosis, which is called metaphase, we should see these chromosomes completely lined up down this metaphase plate equator like you see here. They should all be lined up properly if the pulling forces on either side are equal. Now, I mentioned the spindle checkpoint before, so if we don't do a good job and a timely job of lining these chromosomes up like this, then the spindle checkpoint will be failed, and we will not move on to the next stage of mitosis until we get lined up properly like what you see here. Now, you might be wondering, okay, so if we're pulling from either side, why are the sister chromatids not separating from each other? So why do we line up like this? Why can't we just separate one pair of sister chromatid from another? There's actually a very good reason for that. You can kind of picture for now that these sister chromatids are glued together. There's something holding them together and giving them some cohesiveness that is preventing them from separating just yet. And that's actually a very important part of what we're about to talk about. And like I said before, proper alignment at the metaphase plate is absolutely required for us to pass the spindle checkpoint and move on to the next stage of mitosis, which we will get to here in just a minute. But before we get there, let's discuss that issue that we just brought up. If the sister chromatid pairs are being pulled from both sides, why don't we separate them from each other? There has to be something that's holding them together. So this glue that is holding the sister chromatid pairs together consists of several proteins, and those proteins are not going to be dissolved, meaning the glue is not going to be dissolved until the spindle checkpoint gets passed. So this is a very clever way of making sure that we pass the spindle checkpoint before we make any further progress here. So here is the nature of the cohesiveness, the glue that is holding the sister chromatids together. This is a protein complex called cohesion, and you can see that cohesion is made of several different types of proteins. The names of these proteins are not really that important, but you can actually see that the cohesion protein complex resembles a ring. These rings assemble around the DNA of the two different pairs of sister chromatids, effectively hooking them together and preventing them from dissociating from each other, even if there are pulling forces that are attempting to pull them apart. This is literally the glue that is holding the sister chromatids together. So when we move from the pro-metaphase to metaphase transition, then once we get to the point where we're about to pass the spindle checkpoint, a protein, specifically an enzyme called separase, is going to start to become active inside the cell. Separase is a kind of enzyme called a protease. A protease enzyme is basically a little pair of scissors that is going to cut proteins in half, okay? So separase is specifically going to cut the protein that makes up the cohesion complex. And essentially what that's going to do is dissolve the glue that is holding these sister chromatids together. So once we get to the point where the spindle checkpoint is passed and separase cuts this cohesion ring, now those microtubule pulling forces that we just saw are actually going to pull the sister chromatids apart from each other, as you can see going from left of this figure where the cohesion complexes are intact to the right side of this figure, which is what happens after the separase becomes active and after those cohesion rings are cut. So there are some important details that I left out here. So the timing of separase suddenly becoming active all of a sudden in metaphase, this is a process that's driven by a number of different proteins. You can look that up if you're interested, certainly, but for the sake of general biology, learning about mitosis and the cell cycle for the first time, it is not essential that you look into that just yet. Okay, so the next stage of mitosis after separase does its job and after we separate the sister chromatids from each other, this is called anaphase. To aid in the visualization of this, I have shown you where the metaphase plate was, so you can actually see the progress we are making here in pulling sister chromatids apart from where they were originally. So the microtubules now pulling and retracting towards the centrosome are going to pull the sister chromatids towards each spindle pole, which is where these centrosomes are located, and away from the metaphase plate. The last stage of mitosis is called telophase, and once we accomplish our mission of getting the sister chromatids pulled to either side of the cell, we are going to start reassembling new nuclear envelopes around what's eventually going to be the daughter chromosomes that will be inherited by each daughter cell. And you can see here how each daughter nucleus is now going to inherit a diploid count of chromosomes. So we started with 92, 46 should be in the left cell, 46 should be in the right cell. So after cytokinesis, which is what's going to come next, we should end up with 2 cells that each have 46 chromosomes. The other thing you're going to see here at the tail end of telophase is that a contractile ring that's made of a protein called actin and myosin is going to assemble near the metaphase plate, and it is going to start pinching the plasma membrane. So you can actually see a constriction point where you can really visualize how this big old cell is going to get split in two. After cytokinesis, which results in the division of both the cytoplasm and plasma membrane of the big parent cell into 2 genetically identical daughter cells, we can say that these cells are genetically identical because they are actually retaining the copies of the DNA that were produced during S phase. And you'll recall, we made a big old deal about how that duplication has to be pristine. You cannot have any differences between the DNA after S phase compared to the DNA before S phase. Okay, so that is going to wrap up this review of the cell cycle and mitosis. I certainly hope you found it useful and hopefully interesting. Be on the lookout in the future. I will be recording a similar video about meiosis, which is similar to mitosis, but it is actually a process that is specific for making gametes, sperm and egg, for sexual reproduction. The goal of meiosis is a little bit different compared to mitosis, but once you watch both videos, I think you'll have a good appreciation for the important differences between them and as well as the similarities. Well, I thank you for your attention, and I look forward to you joining me next time. So